So I'm presenting work I've done with Jimmy Ba and our PI, Brendan Frey, on classifying microscopy images with deep multiple instance learning. So the images we typically work with are from high content screens. So here's a sample of yeast GFP protein imaged with such a system. And the type of experiments we can do with this are finding outliers. So in this case, we're looking at different compartments in the cell and how, and we screen them across deletion mutants to see which deletion mutants can cause morphologies that differ from wild type. And we can also look at classification type problems where we want to classify cells into cell cycle stages and see how protein localization changes with the cell cycle. So for most of the talk, I'll be talking more specifically about protein localization. So this is a hard problem for a lot of groups because the patterns are pretty complex and a lot of simple classifiers can't do a good job classifying them. Our group was one of the first to quantitatively assess protein localization on a proteome-wide scale. So this was work that came out in Cell in 2015, and that allowed us to assign a protein, a compartment to every single protein, and also measure its abundance. And then if we conducted this experiment under different conditions, we can see whether localization or abundance would change under that condition, which ultimately gave us a rich data set of functional genomics to work with. And the pipeline people typically use for these experiments involves segmentation, then subsequently extracting features from individual cells, followed by machine learning tasks such as outlier detection, classification, or clustering. So the approach our own group took for classification for the previous paper involved an ensemble of 60 binary SVMs. So you can imagine that's a really complex model to set up, and it's also a complex model to deploy. So even though it worked for that one paper, we've had trouble re-implementing it on new data sets. So ultimately, the challenges with existing methods is that despite advances in algorithms, there's still a lot of groups that are doing this kind of work by eye. So 2 out of the 3 recent proteome-wide screens still assessed localization by eye. And our group, which did it quantitatively, ended up being a huge effort. We required labeling of 70,000 individual cells. We trained about 1,500 models because we used binary SVMs, each with 25 bags. It took probably over 5 years to develop. Like the students that worked on it were gone before I even started my PhD, even though the paper only came out in 2015. And then also it didn't really generalize to new data sets. So pretty much we'd have to repeat this whole process whenever we do a new experiment. So around the time I started, deep learning became really popular. So since 2012, with the first GPU-based implementation of deep learning algorithms have pretty much taken over any challenge related to object recognition and images. They've also been really good at speech recognition and other tasks as well. And the primary benefit of deep learning, in my opinion, for these data sets is that they're actually trained on the raw data, and they're trained specifically for the task that you're working on. So in that case, the features you end up building your classifier on are tuned toward the data set you have, and they're also tuned toward the classification task that you're working with. So because microscopy images are a pretty unique type of image, just taking feature sets that exist for general computer vision problems doesn't necessarily capture all the types of features that we'd want to see in cell-based screens. And convolutional networks specifically have been very popular for images. So they have 2 main benefits. One is that they reduce the number of parameters in the model by having locally connected weights, which are convolved across the input images. Also, pooling layers reduce the dimensionality, typically by half. And ultimately, there's usually a fully connected layer at the end to actually classify the image. So these, since pretty much 1998, have been really good for classifying images. So the problem we want to tackle is how do we use these models for an image like this, where it's a heterogeneous population and there's lots of cells, and we want to do this hopefully without doing segmentation and hopefully without labeling individual cells. So the naive approach, which we've done with the previous paper, is to segment the cells and then label them by eye into different categories. And with that, you can create like a pretty accurate data set to work with. But ultimately, it's really time consuming to do that for every new experiment that you want to do. A second approach is to still segment the cells, but just annotate them with whatever we know about the whole image. So for example, if we know where the protein localizes to, we can just say, okay, we know this protein has these localizations, all these cells have that GFP protein. Let's just assume those are the classes. So this can work well for some categories. So for example, here we can see that in nuclear periphery, it's more or less homogeneous, but then some more transient localizations are harder to label in this way. So what we aim to do is be able to classify the whole image, but still be able to use the multiple annotations. So to do this, we used 2 different frameworks. One is a recent advancement in convolutional networks, which has been called fully convolutional networks, and people have used it mostly for segmentation data sets. So the idea is that you can take a neural network that was trained on cropped images, typically in these cases from the ImageNet data set. So they have fully connected layers, which give you just a prediction vector across all the classes for a cropped image. And if you convert those fully connected layers into one-by-one convolutions, you can take the exact same parameters and apply the same model to a larger image, and you end up getting a heat map for every single class rather than just a single value. So the groups that have used this for prediction have used this as a base, and then they use the pixel level ground truth labels in order to build a segmentation model, but that's initialized with these parameters already. So if we try that with the cell data, we can take a model that we trained on cropped labeled cells, and without doing any extra work, just by converting the fully connected layers to one-by-one convolutions, we can convolve across a larger image, and we can see that in this case, the protein localization is mostly bud-neck, and mostly bud-neck activations appear. The other one that's really active is ghost, which just refers to background. So let's say we wanted to classify this image really quickly. We can just say, okay, which out of these activations have the highest number of pixels activated. There's still some noise, and ultimately you'd get rid of that by further training. Here's a second example that also looks really nice. So this is for a nucleolar protein. So you can see that the area where the cells are is really active for the nuclei feature map. But still, we'd like to train these models without actually having to rely on having the cropped label data set. So to do that, we've turned to multiple instance learning, which is a framework that was actually introduced in 1997. And the idea here is that you only have labels for sets of data points, but you don't have labels for individual data points. And the classic assumption is that as soon as one of the instances is positive, the entire bag gets a positive label. So here's just a toy example where you know that these 2 keychains can open a door, and this one can't. So from that, you can infer that the green key, which is common to these 2 keychains, is the one that you actually need to open the door, which this one's lacking. And then mathematically, typically you have a probability value associated with each instance, and you have an aggregation function g, which aggregates across those probabilities to give a probability for the whole bag. Noisy OR is the classic function used, so that one satisfies that assumption, whereas as soon as one instance is active, then the whole label gets that label. So this is our model. We combine fully convolutional neural networks with multiple instance learning, and we do that by treating each class-specific feature map as a bag, and each element in a class-specific feature map as an instance. And we also design the network such that an element in a class-specific feature map has a receptive field approximately the size of one cell, so that each pixel here more or less represents whether that area has that label or not. In the experiments, we tried different pooling functions to aggregate these probabilities, and then we have a fully connected layer to finally map these probabilities to the labels in the image. So we built a demo just with MNIST digits. In this case, there's scattered digits. We use 0 as a distractor and digits 1 to 9 as samples, which are supposed to represent cells in this case. And after training, pretty much what you would do during training is give an image like this the label 5. And after you train it, you can see that all the 5s are identified in the class-specific feature map. And once you aggregate, that 5 has the highest probability over the whole image. So that's the idea with our model. So rather than have digits, we just have cells. And we experimented with different pooling functions. So we found pretty quickly that the classic ones don't really work in this setting. So noisy OR, ISR, those ones are very sensitive. So they're not really designed to have this many instances per bag. So pretty much right away, they all activate the bag level label. whereas we experimented with other ones such as a generalized mean, log sum exponential, and one that we proposed called noisy AND. This one's meant to mimic the logical AND function in a probabilistic setting. So it's a sigmoid with an adaptable bias that's always normalized to be between 0 and 1. And the adaptable bias is learned per class, so that way if a certain category requires less cells to be activated, that can move around. And here we can see that actually happening for different classes on one of the data sets we trained on. So these are the results we got for predicting whole proteins, so what the protein localization is overall. And we checked the agreement with the original GFP data set, because this one was manually labeled. So our previous paper in Cell has a average, gets a mean average precision of about 0.703, whereas most of our convolutional MIL models do better. We also show that max pooling as an aggregation function really doesn't work in this setting. And then these metrics are similar, just they're only for proteins that have a single localization, whereas this one also deals with proteins that could have multiple localizations. And the pooling function we proposed does the best across the convolutional MIL models. For the yeast data set, we also had the crop cell data from the previous experiment, so we can actually evaluate how well our models do at still segmenting single cells. And here we know exactly what the localization is from the manual labeling that we've done ourselves. And as a second baseline, we took the crop cells and just annotated them with the whole image labels, which was what I mentioned before, kind of produces noisy labels that you wouldn't necessarily want to train with. So in this case, a convolutional net trained on noisy labeled cells does worse on the manual labeled cells, which makes sense. And our MIL models actually do better on the manual labeled cells than on the noisy labeled cells. So to me, that was a really good indication that our models are actually picking up the proper phenotypes for different categories. We also wanted to show that we can use this for data sets outside of yeast. So this is a data set from the Broad Institute, which is publicly available. It's a drug screen for cancer cells with 103 different compounds and 12 that are categorized into 12 different mechanisms of action. So once again, we applied our model with a similar fraction of the data used for training and testing as a previous publication, which used factor analysis for this. And once again, our models do better at predicting the overall mechanism of action. One thing to really note from both data sets is that even if the results were similar, our model takes a lot less effort to implement. All you need is the annotations for the whole image and the whole images. There's no segmentation or labeling done per cell. Whereas both the previous data sets, both the previous approaches required segmentation, feature extraction, and in this case, they didn't label by eye, but in the yeast data set they do. So ultimately, it's a much easier method to deploy and to train. We also know that researchers conducting these screens are still interested in single cell data. So we wanted to see whether we can use these models to localize where the single cells are in the image. So for this, we used some methods that have come out for visualizing features in deep networks. So in these cases, what they do is from a specific feature activation, they backpropagate to the input, and then those gradients are supposed to represent more or less what that feature is looking at within the network. So this paper just uses it for visualization. Here they use it for initializing graph cuts to do weakly supervised segmentation. So in this case, you only tell the model what's in the image, and then through the gradient information, you can actually do the segmentation that way. So here's an example with our data. You can take a query image, pass it through the network. The next step is to pretend all the labels are wrong, and then you propagate those gradients back to the input. And what this really shows is which pixels need to change the most in order to change the label, which more or less tells you where the cells are, because the cells are the objects that are that the prediction of the network is based on. So we can use this for segmentation just by thresholding the gradients and then applying loopy belief propagation to clean up the noise. So it's hard to see because I used purple for the outline, but you can get really good cell outlines with this model. And we can quantitate this, but you can also do it on a per class basis, and you can actually figure out which cells are activating which category. But this is so far just a visualization technique. We weren't able to quantify it. So in summary, we developed a generalization of convolutional networks specifically for microscopy data sets. Our models are easy to train and also easy to deploy. There's no intermediate steps, so you can imagine having an image coming directly from a microscope and then you classify it right away. And I'd like to thank the groups I work with, Charlie Boone and Brenda Andrews, and Jimmy, my co-author, the Frey Lab, and our funding was from NSERC when we had some GPUs donated by NVIDIA. So thank you.